Hello everyone, welcome to Get My Class. So let's solve this question here. It says the technique of DNA fingerprinting includes the first one says isolation of DNA, the second one says detection of hybridized DNA fragments by autoradiography, third one says separation of DNA fragments by electrophorus, and option 4, the fourth step, it says hybridization using labeled VMTR probe, and the fifth one says digestion of DNA by restriction endonucleases. Sixth one says transferring or blotting of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon. And they've given various options, that is A, B, C, D, and they have mentioned a different sequence of these steps. So what are we supposed to do? According to the question, it says the technique of DNA fingerprint printing includes. So they're talking about the various steps of DNA fingerprinting, and we have to look for the correct order and choose the correct option. Okay. So before we actually look for the correct order, first of all, we have to understand what is DNA fingerprinting. DNA fingerprinting is nothing but it's a procedure by which we can find out the specific repeated sequences of a particular person's DNA. Okay, so here it is basically to you can say you can look for specific sequences of the DNA. Okay, and this is to check whether the sample what they have got, does it belong to a particular person or not. Okay, so that is the main usage of DNA fingerprinting. Apart from that, it is also utilized to check whether if there is a baby or there is a child whose parents are unknown, then they can look for or they can perform this DNA fingerprinting of the child as well as parent, and they can find out the real parents. Apart from that, criminal scenes, or you could say the samples collected from the crime scene, they can be considered for DNA fingerprinting, and further it can be analyzed and looked for the culprit. Okay, so let us see. So let us understand the actual procedure of DNA fingerprinting. So as the name indicates, it says DNA fingerprinting. First of all, we need the sample, and the best sample for DNA fingerprinting is the blood sample. And from the blood cells, we will isolate the DNA. Okay. Once we isolate the DNA, we will digest the DNA to get fragments, because we cannot put entire DNA content onto the gel electrophorus to run it, because it'll not move, because the entire genetic material will be very, like, the content is huge, right? So what do? We use restriction endonuclease enzyme or restriction enzyme, which will cut the DNA at specific sites and give rise to different size fragments. And these fragments, or this sample containing fragments, will be loaded on, this is called as the gel electrophoresis. This procedure is gel electrophoresis. Okay, gel electrophoresis, where by using current as well as on this agar gel and with certain chemicals, it will be allowed to move from, you can say, from the negatively charged electrode to the positively charged electrode, as the DNA fragments are already negatively charged. So they will definitely move towards positively charged electrode. And that's how they form different bands. Okay, once they form different bands on these, on the agarose gel, then they will transfer that agarose gel, or you can say transfer those bands from agarose gel onto either nitrocellulose sheet or nylon sheet. This is called as blotting. And since here the transfer is happening of the DNA bands, we call it as southern blotting. What do we call it as? Southern blotting. It is southern blotting. Okay, southern blotting. You also have northern blotting and western blotting, where northern blotting is used for RNA and western blotting is used for proteins. Okay, but here, because the DNA fragments are transferred, it is called as southern blotting. Okay. Further, what happens? Like, for once the DNA, you can say the bands are transferred onto the blotting sheets, or you can say nitrocellulose sheet, then what do? They treat those bands containing sheet with probes. Okay, so they use probes, tiny probes, whose sequence they already know. so as to find the presence of complementary sequence on these bands. Okay, so they are called as probes, and these probes are nothing but the VNTRs. That is, you can say variable nucleotide tandem repeat sequence, because these are specific for specific person. Okay, so that is why it is very important to study about these tandem repeats. Okay, and further, then what happens? Then they will take that sheet and then they will wash it. Okay, and so as to remove any excess probe, and then at this stage they will use the radioactive probe is bound to the DNA pattern on the membrane. So then when they remove the membrane, you can see that the radioactive probes, because these probes are radioactive, they will be helpful in identifying the specific bands of the DNA of the sample, because they have the capacity to emit radiation because they are radioactive. Then this sheet will be subjected to X-ray, okay, and further X-ray film will be obtained where you can get the picture of, or you can say exact pattern of the band. Okay, so let us see. So the first one says isolation of the DNA. That is absolutely right. This is the first step. Okay, the first step is what? This is the first step, isolation of the DNA. Then it says detection of hybridized DNA fragment with autoradiography. So that happens at the last, right? That is here. That is the 10th step. Okay. Hence, this is the last step. So we will keep it aside. Then comes separation of DNA fragments by electrophoresis. So before we separate, we have to digest them with endonuclease. So this will be third step. And let us see, hybridization using labeled VNTR probe. So that also comes later. Digestion of DNA by restriction endonuclease. Yes, this is the second one. So first you isolate, then you use a restriction endonuclease to break them into fragments, and then you run electrophoresis, right? And then transferring or blotting of separated DNA fragments to synthetic membrane or such as nitrocellulose or nylon. This is the fourth step, as you can see. So this is first step, this is second, and then this is third, this is fourth, yes. And then what comes? Then you have the probing, the fifth one. This is the fifth one, right? And lastly, you get the sixth one. So here, this entire process can be considered as the sixth one. Okay, so it should be one, then it, okay, let me write here, one, then you have five, then you have three, okay, then the fourth one is six, then you have fifth one as fourth, and you have sixth one as the second. So 1, 5, 3, 6, 4, 2. Where is 1, 5, 3, 6, 4, 2? Yes, option A says 1, 2, so it is wrong. Whereas B says 1, 3, 2, which is again wrong because we have 1, 5 initially. C says 1, 5, 3, 6, 4, 2. This is absolutely right. Option D says 1, 3, 4, 5, 6, 2, which is again wrong. So which is the correct answer? The answer is option C. So let us conclude. The answer is option C. I hope you understood the explanation. Thank you.